I wonder what's in that soil.
Well, it might not look like much, but there's actually a whole world down there. For example, the soil-dwelling microbial Streptomyces avermectins lives down there.
Oh yeah, in 1978, 3 scientists in Japan were able to isolate a really useful molecule from it, avermectins.
Yeah, Streptomyces avermectins uses avermectins as a form of chemical warfare against other soil-dwelling bacteria and parasites that compete for resources.
And who knew that humans would find so many uses for them too?
Humans really have found many uses for avermectins, applying them in agricultural, veterinary, and medical fields, generating worldwide sales of $850 million. It's invaluable to understand the biosynthesis of avermectins and increase their production. In Streptomyces avermectins, avermectin is synthesized through a complex pathway. It begins with the synthesis of avermectin A-glycone, catalyzed by polyketide synthases. The A-glycone is then modified. The most important step in this modification depends on whether AvC and AvD genes act, which determine the A or B and 1 or 2 series. Finally, the A-glycone is glycosylated with sugars. The triggering of this pathway is not completely understood, but scientists are working on ways to increase their production. These methods include utilization of mutagens and optimization of fermentation conditions. Recently, genetic engineers have tried their hand at increasing production, finding that AvT gene overexpression increases production 1.2-fold, and deletion of AvM genes increases production 3.5-fold. The newest forms of increasing production include the use of nanosecond pulsed electric fields.
Now, let's take a look at the structure of avermectins. First, notice the purple 16-membered macrocyclic lactone ring. This identifies avermectins as a member of the macrolide antibiotic family. Second, notice that carbon-13 is diglycosated. Derivatives are identified by dividing into 4 types: A1A, A2A, B1B, and B2B, and then various further subtypes by different substitutions at R1, R2, X, and Y positions shown here in blue.
Before we talk about the mechanism of avermectins, let's discuss some basic physiology. For Audrey to make her muscles move, she needs an action potential to happen. For an action potential to happen, we need certain ions like sodium, potassium, and chloride to move across the cell membrane. When these ions move across the cell membrane, they change the likelihood of an action potential to happen.
Isn't that how avermectins work?
It is! So let's go take a better look.
Shown here is the neuromuscular junction, which is the site for the transmission of action potential from nerve to muscles. Normally what happens is glutamate neurotransmitters are released from the interneuron terminal into the synaptic cleft. Glutamate then moves to bind to GABA receptors, causing a conformational change and opening up the channel. When the channel is opened, chloride ions are free to move through the channel and into the motor neuron terminal. When this happens, the cell becomes more polarized and decreases the likelihood of an action potential to happen. This means that no signal will be sent to the muscles to move. When avermectin comes in to act as an antagonist to the receptors, avermectin binds to the receptors and allows chlorine to move into the cell. The difference is that avermectin remains bound to the receptor longer than glutamate and will increase the duration of the hyperpolarization. This causes paralysis and death to the organism. Humans, however, don't have to worry too much about this mechanism because the structure is highly polarized, meaning it can't cross the blood-brain barrier.
With integrative pest management, I use a combination of monitoring, cover crops, crop protection products, and more.
Hey, what are you watching?
Just an ad about pesticides.
Oh, have you ever wondered how they work?
No, not really.
Yeah, me either. But let's go find someone to tell us anyways.
Yeah.
Hey, it's Audrey.
Hey.
Do you know how pesticides work?
Actually, yes. I love pesticides. I'm sure you've heard about avermectins. Well, one of the most common derivatives, abamectin, is the main ingredient in commercially available pesticides. So, I'm sure you already know about how abamectin causes paralysis. Well, this paralysis leads to starvation and the death of the pests. It is especially effective because it is not harmful to humans. The GABA receptor exists in the blood-brain barrier of humans, and abamectin cannot penetrate it. This isn't the case for the pesticide attacks. but it's still a very toxic derivative, so it's sprayed in low concentrations. And despite this, it's generally environmentally friendly because it has many biodegradation pathways and it's very immobile in the environment.
I'm itchy, and I'm blind.
Oh no, you have river blindness.
Oh no, am I gonna die?
No, don't worry. I have ivermectin. Here you go.
I'm cured.
Did I hear ivermectin? Ivermectin is one of the most commonly used derivatives of avermectin. It is the only derivative available for over-the-counter use in humans, and it was the first FDA-approved drug for the treatment of onchocerciasis, aka river blindness, a parasitic disease. Since its first usage, it has been expanded to treat many other parasitic diseases like head lice and scabies. Ivermectin is also currently in clinical trials for COVID and malaria treatments. For malaria, the mechanism isn't quite clear since the parasite responsible lacks the ivermectin target. For COVID, toxic neurological side effects have been observed due to inflammation of the blood-brain barrier. This toxicity is the reason that COVID clinical trials are no longer underway. To describe a few more uses of ivermectin, let me hand it over to my veterinary friend.
I use ivermectins every day for animal treatment. They help control common parasite infections like heartworms, ectoparasites, and microfilariacides. Controlling these parasites is important for livestock health care. Due to crowded populations, that can lead to increased spread of infection and death. Oh no, stop!
Why?
Ivermectins are toxic to dogs due to a genetic mutation that can lead to death.
I didn't know ivermectins were toxic.
Yeah, some of the side effects include hypersalivation, bradycardia, and blindness.
And what are the other negative symptoms?
I don't know. Let's go outside and find out.
Like I said earlier, ivermectins are a generally environmentally friendly molecule. However, there are some negative side effects. For example, some soil microbial communities face metabolism imbalances that negatively affect the health of the community. Despite this, ivermectins have a relatively short half-life in the environment, and that depends on the climate it's in. In temperate climates, it can last and be active in the environment for up to 2 months, but in Mediterranean climates, it's only active for up to 6 days. Overall, ivermectins are a very useful and interesting family of molecules. Thanks for watching, and I hope you learned a lot.